Hello, I'm Dr. Jeremy Graff, Chief Scientific Officer and Senior Vice President for Research at Biothera Pharmaceuticals. What I'd like to take you through in this module is the ability of Imprime PGG to drive an integrated anti-cancer immune response that potentiates the efficacy of immune checkpoint inhibitor therapies for cancer. When we think about immune checkpoint inhibitors, we recognize that these inhibitors can work and work very effectively, but do so in a minority of patients. 15 to 30% of the patients treated with these drugs respond very well. In those patients who respond to single-agent checkpoint inhibitor therapies, we can recognize that there is an ongoing T-cell-based anti-cancer immunity. This is evident by the infiltration of CD8 and CD4 T cells, by PD-L1 positivity on the tumor cells, as well as myeloid cells that inhabit the tumor bed, and by an adaptive immune signature. For checkpoint inhibitor therapies to be truly effective, we need multiple elements of the anti-cancer immunity cycle to be in force. First, we need a sufficient antigen debris field. These antigens must be sufficiently foreign to be recognized as non-self and be presentable. Second, we need a permissive immune microenvironment, one that is characterized by M1 state macrophages rather than M2 state macrophages, one that is characterized by reduced or differentiated myeloid-derived suppressor cells. Three, we need effective antigen presentation. Primarily, we think of active mature dendritic cells, but other cells like M1 state macrophages may also participate in presenting antigens with the appropriate co-stimulatory signals to T cells to drive T-cell-based anti-cancer immunity. Imprime PGG is a novel therapeutic currently in phase 2 clinical studies in concert with checkpoint inhibitors. Imprime PGG is a beta-1316 glucan isolated from the cell wall of the yeast Saccharomyces. It acts as a pathogen-associated molecular patterning molecule, or a PAMP. PAMPs are the critical non-self danger signals that our immune system, particularly our innate immune system, uses to become activated in response to a normal pathogen. As a consequence, Imprime acts as an immunological ignition switch. It allows for antigen presentation, repolarization of the immunosuppressive tumor microenvironment, and it activates tumor cell killing. Importantly, Imprime is systemically administered and works in people as an immune complex. In the next module, we'll discuss the importance of that immune complex. Imprime directly activates tumor cell killing, thereby enhancing the antigen debris field in the tumor bed. Imprime repolarizes the immune microenvironment, enabling macrophages to take the M1 polarization state, driving the differentiation of myeloid-derived suppressor cells, and ultimately allowing for the activation of antigen-presenting cells, the maturation of the dendritic cells and the M1 state macrophages that then can effectively communicate with T cells to drive T-cell-based anti-cancer immunity and to close the loop on the anti-cancer immunity cycle. In our phase 2 trials, we will be able to assess the impact of Imprime on each of these elements of the anti-cancer immunity cycle and in driving T-cell-based anti-cancer immunity. For additional information, please visit our website. Thank you for your attention.